lesson we're going to look at DNA transcription. Now this is the classic shape of DNA. You've probably come across this before. But the one thing you've got to realise is that what DNA does, DNA codes for proteins. So proteins are the basic building molecules of the cell, they're what enzymes are, so they're really, really important. And DNA, the code in DNA, dictates to the cell what proteins are going to be made. A bit of background now about DNA. First of all, DNA consists of two strands wrapped into a double helix, you can see here. The two strands are linked by pairs of bases, so between the, if you like, the uprights of a ladder, you've got these rungs, and these rungs are pairs of bases. There's one here and one there, and there are four bases, what's called adenine, thymine, cytosine and guanine. And the way it works is that adenine always links to thymine and cytosine always links with guanine. So for example, if green is adenine, that would be thymine, adenine, thymine, adenine, thymine and so on. Okay, so adenine always links with thymine and cytosine always links with guanine. Now the marvellous thing about these things is that the bases, which are these rungs here, are read in threes or triplets. So this here would be a triplet because it's 1, 2, 3 bases. And each triplet codes for a particular amino acid. And that's really important. As you know, proteins are made up of amino acids. So reading from the bottom, this could be cytosine, cytosine, thymine. There's a triplet there. Okay, and cytosine, cytosine, thymine actually codes for the amino acid cysteine. Now there's a problem, and it's quite a big problem, in that DNA, which is the genetic code for making proteins, the instructions, if you like, are found in the nucleus of the cell. So the instructions are here. Now, the site where proteins are made is called the endoplasmic reticulum, and that's also in the cell, but it's over here. So the question is, how does genetic code get from the nucleus here to the endoplasmic reticulum here, and also how are the proteins assembled from amino acids? Thankfully, there's a very, very clever solution. Now, the synthesis of proteins by joining amino acids together depends upon two very special molecules. The first one we got is called messenger RNA, or mRNA for short. Now, we've talked about DNA is deoxyribonucleic acid. mRNA is ribonucleic acid. It's a single strand. It's not a double helix, it's a single strand. So mRNA is a single strand ribonucleic acid. Also, we've got a second very special molecule called transfer RNA, or transfer ribonucleic acid. And again, that too is a single strand, but it's been twisted. Some people liken this as sort of a hair grip. And at the bottom here are very important binding places. Now, there are two very special processes that involve these two molecules. The first one we'll look at today is called transcription. The second one we'll look at another video is called translation. So let's look first of all at transcription. So first stage of protein synthesis is transcription. Here we have the nucleus. This is the pink bit here is the nucleus of the cell. The blue bit is the cytoplasm of the cell. And here is an exaggerated size bit of DNA. Now the first thing that happens is an enzyme. Now enzyme is RNA polymerase. How do you know it's an enzyme? It's got ASE at the end. That attaches to DNA at the beginning of a gene. Now a gene is a section of DNA. Okay, a gene is a section of DNA. And what's going to happen is we're going to be copying the bases from that gene. Now, second point is that hydrogen bonds between the DNA strands break, exposing some bases. That should be strands, by the way, not stands. So these rungs, if you like, break apart. The hydrogen bonds are broken, and what they do, they expose the bases. So we've got cytosine, thymine, cytosine, cytosine, thymine. Those bases are being exposed by the RNA polymerase, if you like, unzipping the DNA. Now, we've got two parts here that could be templates, but only one of the strands is used as a template to make the messenger RNA copy. So we've got this template here, CT, CCT, and then three nucleotides. these container bases line up alongside the exposed bases with the complementary pairing. So cytosine always links with guanine, thymine, adenine, cytosine, guanine, so on. Now the one exception here we need to point out is that messenger RNA doesn't contain thymine. Messenger RNA contains another base called uracil. So U there replaces T. Okay, so if we had an A here, which we will have later on perhaps, you would replace that with, so that would be binding with a U, a uracil, as opposed to a thymine. The RNA polymerase joins together the base pairs to form messenger RNA molecules, and you can see here the messenger RNA molecules starting to form. So the RNA polymerase moves along the DNA in this direction here, and as it does so, it exposes more bases. Look, there's what we talked about. There's adenine there, this time combining with uracil. So it goes along this bit here, and it separates the strands, so more and more bases can join up to form a messenger RNA chain. As it does so, behind it, the hydrogen bonds behind the uncoiled DNA strands reform, coiling back into double helix, so it gets back to what it was before. So RNA polymerase stops making messenger RNA when it reaches a DNA stop signal. What does that mean? Well, it means that certain triplets actually code for a stop. They say stop making the messenger RNA here, and also detach the messenger RNA from the DNA. You can see here messenger RNA strand leaves nucleus, and then it begins its journey to the endoplasmic reticulum. So as you say, messenger RNA, now a strange word here, eukaryotes, explain this in a second, moves out of the nucleus through a nuclear pore. Nuclear pore is like a tiny hole in the nuclear membrane through which the messenger RNA can travel. And what it then does, it attaches in the endoplasmic reticulum to a ribosome. We'll come at that in another video. Now, some important things here I need to point out. This is a summary here, just showing you all the stages that take place when we're making messenger RNA. Now, transcription differs whether we're talking about eukaryotic cells, strange line that, or prokaryotic cells. Prokaryotic cells are very, very basic cells. They're like the early cells in evolution. So things like bacteria are prokaryotes. They don't have membranes around the nucleus, for example. They don't have membrane-bound organelles. But eukaryotes, we are eukaryotic cells, and we have membranes around the nucleus, around other organelles in the cell. Okay? But it differs in terms of transcription. Eukaryotes have got both introns and exons. That sounds rather strange. Let me explain. Introns are sections of DNA that don't appear to code for anything, whereas exons are the coding sections. Some people refer to introns as being junk. I'm not going to say that here because it might be insulted, but introns are areas of DNA that are not needed, whereas exons are the important bits. But both are copied into the messenger RNA during transcription. Messenger RNA strands containing messenger RNA are called pre-mRNA, another term for you there. We then have splicing, like what we might have done years ago for videotape. And what happens in splicing is all the introns, all the non-coding DNA is removed, and then the exons are shuffled together to form the messenger RNA strand. And this takes place before mRNA leaves the nucleus. In prokaryotes, there are no introns or exons, just exons if you like. So messenger RNA is produced directly from the DNA without splicing having to take place. Right, that's the end of this video. I hope you understand more now about the DNA transcription, and I'll be back to look at DNA translation very shortly. So bye bye for now.